girls. So what we're going to do is we're going to go over basically a cell in action. And I'm going to show you how insulin is made and then how it's released. And I'm going to kind of go over some parts that we've looked at in terms of a cell. So what I'm going to do is I'm going to try to tie in together a few concepts, okay? All right, so the first thing we're going to do is ask a really simple question. Does anyone know what insulin does? We talked about this yesterday. It helps the body take up sugar, okay? Now, this is the cell that makes it. So I'm going to give you its name. This is called a beta cell. All right? It's called a beta cell. And this is the cell that makes insulin, all right? This is a cell, basically. But what I don't have in this diagram, I don't have all of the organelles, okay? So, and some of them, I'm not even going to draw them because we kind of know what they look like. And just, it'll take up a lot of space. So, first question is, when does a beta cell know to release insulin? What do you think the answer would be for that? What do you think the answer would be when this happens? So the answer is when when it senses glucose. So you eat sugar, beta cell senses glucose, and then it releases insulin. What we're going to do is we're going to take a look at basically how this works, okay? So what you're seeing here is the outside of a cell, which you know is called the cell membrane. And you'll notice that there are things in the cell membrane, okay? And the cell membrane is something that controls what goes in and out of cells. And you can see from this picture that one of the things it controls is the movement of this, okay? Glucose. So this is the doorway for glucose, all right? If this cell did not have that doorway, then the glucose would not be able to come in. All right, does that make sense? Easy enough so far? All right, now, you'll notice in this diagram, once the glucose comes in, something happens to it. And we're not going to go into detail as to what happens. That's more like senior biology stuff. But something happens to it, and there's a protein that modifies the glucose. And here's his name. Okay? It's got a nice name. Now, I'm going to ask you a really simple question. This is a protein that stays in the cell. So what type of ribosome made it? Which one? We learned about two types of ribosome, free or bound. Very good. So what made this was a free ribosome. Okay? So a free ribosome made that molecule called glucokinase. Now, let's pause. Yeah, you asked me a good question. So glucokinase is a protein. And remember I said to you that cells need to make proteins. You see these doorways? Those are all proteins. Okay? So that's actually a very good point. Who made those doorways? So who made these? It wouldn't be the free ribosomes. It would be the bound ones. Remember, the bound ribosomes make things that either go to the surface of the cell or they leave the cell, and the free ribosomes make the proteins that actually stay in the cell. So the big difference there. Insulin, then, would be an example of another thing in this diagram that actually gets made by a bound ribosome too. These yellow dots are insulin, okay? And you can probably see that on your I think you can see it on your diagram. So these little yellow dots are all insulin, and they're leaving the cell. So these are proteins that are leaving the cell, so they're made by bound ribosomes. Okay, and then we talked about the difference between those two types. Now, one thing that's really cool about a cell is it's like you got to think of it like a system, and all of these parts working together. All right, and we talked about the mitochondria. Do you remember what the mitochondria does? What does the mitochondria make? It makes what? Starts with an E. Yeah, mitochondria makes energy. So the energy is actually this. These things called ATP represent energy, and they come from the mitochondria. Okay? So these things come from the mitochondria. Now, if you look at this diagram, and it's kind of hard to see by just looking at this diagram, but the energy actually does something to this molecule right over here. What it does, it actually closes it. So what happens when it closes? So this is actually a doorway, and we'll talk more about these types of doorways later on. But this is a doorway that allows positive potassium to leave. So let's think about chemistry here, all right? What would happen if we close the doorway and the positive potassium can't get out? What happens to, and you got to think of it, why is it allowing this potassium to leave, right? So if you let it go, you're losing positive ions. What would be the opposite of that? Would be if you keep the positive ions. Imagine you start to accumulate positive ions on the inside. Okay, so you start to accumulate this. What happens to the inside of the cell? It becomes more positive. Okay, this is really important. In grade 12, you'll learn about how important that is. This is how your nerves work. Okay, but the change in the charge, this is grade 9 chemistry now, right? The change in charge actually affects another protein. It affects this one. This doorway is called a calcium channel because it actually lets calcium in, and it's controlled by a voltage. When the voltage goes in a positive direction, that doorway opens. So let's recap here. This doorway, this one over here, allowed glucose to come in, number one. The glucose then gets converted to energy. The energy is used to close this doorway, which changes the voltage, which then opens this doorway. Alright, okay, so why do we need calcium? Well, calcium is actually an important molecule in a process called exocytosis. Now, we haven't studied exocytosis, but just by looking at its name and seeing as to what's happening over here, what do you think exocytosis means? So it refers to the exiting of a molecule. And here it is right in here. So in these little packages, and I think you know what these packages are called. They're called vesicles. Okay, is insulin. The insulin is in here. And the insulin will leave when it detects a high level of calcium. So now you can see like a domino effect. The glucose comes in, gets converted to energy, the energy by the mitochondria, the energy closes the potassium channel, the voltage changes, calcium comes in, that triggers the release of insulin. Alright, now let's see if we could tie in together some other organelles. So here's a question. The instructions to build insulin, to build these doorways, where do they come from? Where do the instructions come from to build all of these structures? And the answer is here, DNA. And you know the DNA is found in the nucleus. Now, the thing is, we talked about these structures either stay in the cell or they leave the cell. So the DNA has to be converted to another molecule and then read by the ribosomes, right? And if it's free, they stay in the cell, and if it's bound ribosomes, they leave the cell. Now, if you notice, there is a problem though. The problem is this. The problem is, if you look at what's happening in the diagram, you'll spot that the cell will actually get bigger as these things leave. So what's happening is, as the vesicles get to the surface of the cell, the cell membrane actually gets bigger, and the cell would get bigger. But the thing is, if you look at a cell, it doesn't get any bigger. So what's happening then? So that means that the opposite must be happening. We must have vesicles forming that are going into the cell that are getting broken down. And the question is, who's breaking down the extra membranes? Because if we don't take away those membranes, the cell's going to get bigger and bigger and bigger. Who's taking away the extra membrane? Who's digesting the membranes? Who's breaking it apart? Who's recycling those components? Starts with an L. The lysosomes. All right? The lysosomes are breaking down this extra membrane. So you see how we've tied together a lot of parts, right? And I did miss two things that we'll talk about really briefly now. Okay, so the lysosomes digest that extra membrane. Now, the other two things I didn't mention is the rough ER and the Golgi. So it's the last thing I'll mention. The rough ER, if you remember, is the ER attached to ribosomes. So the rough ER is the part of the cell that it makes the proteins that do what? They leave the cell, okay? Because they have the bound ribosomes. What does the Golgi do? Now, the Golgi does something really interesting. The rough ER sends insulin to the Golgi, and you're not seeing the rough ER, you're not seeing the nucleus, you're not seeing the Golgi in this diagram. But the rough ER would send a vesicle with insulin in it to the Golgi, but it's actually not called insulin, and I'm not going to give you its name, because if I give you its name, you're gonna just fixate on the name. I'm going to just call it inactive insulin. Okay? It's an inactive version. What the Golgi does, the Golgi actually changes the insulin to an active version and leaves it sitting in the cell until glucose shows up. And if you remember, that's what started the whole thing. What triggered the release of the insulin was the arrival of the glucose. The Golgi just prepares the insulin and makes it active, and it just sits there until the beta cell is ready to release it.